Welcome to the Gillett Health mini Q&A podcast about the androgen receptor, tadalafil, L-Carnitine, and many other things.
So to start us off, what is the androgen receptor and where is it located?
The androgen receptor is a protein and it's located all throughout the body in different levels and different tissues. And this protein binds to various different androgens such as testosterone, such as DHT, and it has many different functions and roles. But at the end of the day, what it does is it goes into the nucleus and causes gene transcription from the androgen receptor gene.
Interesting. So what is this concept of androgen receptor sensitivity that we keep hearing about? Good androgen receptor sensitivity, bad androgen receptor sensitivity. Could you talk about that?
Definitely. So there's a lot of misconceptions when it comes to androgen receptor sensitivity. There's a few ways to think about it. One way to think about it is the density of the androgen receptors that are in the cytoplasm itself. So if you have more androgen receptors, you have more places or more targets for your testosterone and other androgens to bind to, and you have more gene transcription that way. There's also true sensitivity, so I don't really consider androgen receptor density as sensitivity. That's just the density. It's the same thing, quality versus quantity. So density, you have more quantity. As far as the sensitivity of the receptor is a protein like many other things, and certain proteins don't fold as well. So one common concept that I talk about is the concept of CAG repeats. And when you have a lot of these CAG repeats, the protein does not fold quite as well, and your androgen receptor literally is less sensitive.
Yeah, that's a really good way to put it. The quantity versus quality analogy, I really like that. So what are some of the implications for how many CAG repeats somebody has with regards to goals like muscle building or even some disease states like prostate cancer?
It's a good question, and it's something that not a lot of people think about. So I think this is going to be one of a concept that is hopefully talked about more and more as time goes on. With very few CAG repeats, you tend to be more sensitive to androgens, and you can be at risk of things like breast cancer, prostate cancer. If you have a lot of CAG repeats, then you're not as sensitive. So one way to think about it is if you have, by the way, the average number of repeats is around 19. So people have what's called a reference genome. In the reference genome, that's the average person. There's nobody with this exact genome, but it's just the average person. And if you have, for example, some countries, I believe Romania has 26 CAG repeats on average. So these people would perhaps be less prone to certain types of cancers, less prone to hair loss, but more prone to sarcopenia, muscle wasting, and other diseases. So you win some and you lose some.
Right, so like with most things, it's best to not be on completely one end of the spectrum as opposed to the other. There's always a balance there. Yep. So there's several mechanisms for things that can potentially affect the androgen receptor, more so the quantity of the androgen receptor. So one thing that gets talked about a lot is L-Carnitine. Could you talk a bit about some of the mechanisms there, including its effect on the androgen receptor?
Yeah, so L-Carnitine is technically a peptide, a very small peptide of only 2 amino acids. So peptides are just strings of amino acids to make up a protein between about 2 and a couple hundred amino acids. So L-Carnitine is one of the smallest peptides. It's tied for the smallest peptide possible. So L-Carnitine is a shuttle, and it shuttles fatty acids and helps with nutrient partitioning. So it can move your fuel into your cell or into your motors, mitochondria, the powerhouse of the cell, and allow you to utilize it more. That does a few things. It allows you to potentially have more energy, which lets your body be more efficient with burning energy, which subsequently downstream to that is going to end in less muscle protein degradation and more muscle protein accrual, which is an endpoint that many people are very interested in.
That's interesting. It sounds like you are literally burning more fat and in a more anabolic state if you have a higher level of carnitine.
Yeah. Carnitine's one of those things similar to creatine, where it's hard to get an optimal amount in your diet.
Right, it probably depends on the bioavailability, what foods it's eaten with, and it just becomes simpler to supplement with something like that. So as far as its effect on the androgen receptor, what do we see there in the either in vitro or in vivo or even in human data?
There's been pretty good studies both with rats and humans for L-Carnitine, the most within the last couple decades. And you see an increase in the density of the androgen receptor in the cytoplasm itself. You also see an increase in the mRNA activity of the androgen receptor. mRNA is the messenger RNA that is kind of it's in between the androgen receptor gene and then downstream to the androgen receptor. And you also see more androgen receptor gene activity itself. So there's more androgen receptors and there's more activity of the receptor.
And the net result of this is probably what most people are interested in, is ultimately driving more muscle protein synthesis for hypertrophy. You hear about carnitine and TMAO levels. Is this something that people should be worried about?
They should be a little bit worried about it. Just worried enough to where if they're on a high dose of carnitine to check their TMAO. Not so worried that it stresses them out and scares them away from it. It's one of the safest, in my opinion, it's one of the safest choices for performance enhancing, both for ergogenic aid and for bodybuilding, muscle protein accrual aid.
Yeah, I like the comparison you made with creatine earlier, because that's another supplement that we know has, vast data and safety and reliably improves athletic performance. So L-carnitine being another ingredient you can get in your diet, if you will, it's kind of on the same level there. So one of our top questions is about L-carnitine, and specifically, what are the doses, how much is safe to take, how much are people taking, and what do the studies show is actually beneficial?
This is a very common question, for sure. So there's a couple ways to take L-carnitine. You can take it orally, where it's usually about 10% bioavailable. You can also inject it, usually in a muscle, since when you're prescribed injectable L-carnitine, it comes in an aqueous solution. And for that, you usually aim around 500 to 1,000 milligrams. For oral ingestion, depending on the person, anywhere from 1,000 to 3,000. It is important to check your TMAO if you're on quite a bit, especially if you're also on choline or choline precursors, as that's a potential carcinogen that oral L-carnitine can raise that injectable L-carnitine does not.
So for our next question, nutrient timing around exercise, how does that affect muscle protein synthesis, and does that affect the androgen receptor at all?
Yeah, so there's kind of two ways to look at this timing, or the timing window. One way is to ask yourself, is there a clinically significant anabolic window? It doesn't matter if it's 20 minutes or an hour, and there's not really a clinically significant anabolic window, at least for 99% of people. However, if you're looking at nutrient timing when it comes to L-carnitine's effects, it's pretty profound. So you can look at L-carnitine in relation to no L-carnitine, placebo if you will, the control group really. So L-carnitine significantly better than the control, and then with fluid intake or water after you work out, you time it to just after, and you have significantly more activation of androgen receptor and better muscle protein, so less degradation. And then if you add food into the mix, you have even better, significantly better than the other two groups. So on paper, it matters, and if you're going to take L-carnitine, then it would be wise to consume nutrients after your workout.
Yeah, absolutely. It's really interesting. I think looking at nutrient intake as a whole, what I would tend to look at is, what are you doing on a weekly basis on average, as opposed to, what are you doing 20 minutes after you exercise and then neglecting the other 98% of the day?
Yeah, definitely. And the day in general is not a magic window, so things don't reset at midnight and then you're magically in a day where you can have, you're like, oh, this is a surplus or a refeed day. Sometimes it's more like a period of 3 days or a week where that's your surplus or deficit, because you can have significantly more or less nutrient shifts over that time. There's nothing magical about a day.
Can you tell us about CAG repeats in the context of some disease states and things that we see that are hormonally driven, like androgenic alopecia or polycystic ovarian syndrome?
Yeah, if you look at your number of CAG repeats, which by the way, if you're a male, you have only one chain of repeats. So you have what's called autosomal chromosomes and sex chromosomes. Sex chromosomes are your X and your Y if you're male, and two X's if you're female. The androgen receptor gene is on the X chromosome, which some people think of as the female chromosome, but your androgen receptor gene is on that chromosome. So for guys, obviously you can have different single nucleotide polymorphisms. So you can have a variation in a couple different polymorphisms of the gene, but you're only going to have one string that has CAG repeats. So for a female, you can have, let's say you have one of your X chromosomes has 15 repeats, pretty sensitive. One has 25, not very sensitive. If you have PCOS, or if you have other like insulin sensitivity, resistance, things like that, you're more likely to have a methylation or an epigenetic inactivation of your X chromosome with 25 repeats. So the one that is more sensitive to androgens is more active, which makes sense because part of PCOS is androgen dominance, and that can lead to anovulation, which is no ovulating, and that leads to infertility. So that is a potentially clinically significant thing to address. From the male side of things, androgenic or androgenetic alopecia, the same thing. If you have fewer repeats, then you're more likely to have weaker androgens activate that androgen receptor gene in your follicle and lead to follicle loss and eventual stem cell movement out of the hair follicle. So you might, if you have, let's say if you're a guy and you have 13 repeats, you'd think that potentially that could be good for lean body mass, ergogenic aid, but potentially very bad for androgenic alopecia.
Yeah, that's interesting. And I'm just speculating here, but we know that males are much more prone to color blindness, which is another X-linked disorder, and much more prone to androgenic alopecia. So I wonder if there's any studies in that general sphere. Do you have any comment on that?
I'm not sure if there's studies linking androgenic alopecia to color blindness itself, but there is, I'd say, like a cultural phenomenon where people know that a lot of times you look at your mother's father, which I think is interesting, because you get your X chromosome from your mother if you're a guy, and she got one of her X chromosomes from her father. So if you look at your mother's father, there's a 50% chance you got your X chromosome directly from him, which has huge significance when it comes to whether or not you'll have that trait.
Yeah, that's quite a large percentage. It's a 50-50 shot.
Yeah. Let's chat about testosterone and other androgens and how they affect the body. So most people know that there's testosterone, which is thought of as the main male hormone, but there's many other hormones like dihydrotestosterone or DHT, DHEA or dehydroepiandrosterone, and even other ones like androstenedione, and non-bioidenticals like oxandrolone, which is Anavar, and many other anabolic steroids that do bind with the androgen receptor. In regards to that, let's talk about how they work in multiple ways throughout the body.
Yeah, so there's certainly a number of different hormones that have androgenic effects. You have your hormone that's mainly thought of, testosterone, as being the male sex hormone, but then, as you mentioned, that is actually converted into DHT by an enzyme called 5-alpha reductase, and they have very different effects. So for example, testosterone is a potent driver of muscle protein synthesis, and why that happens is you have testosterone goes into the cell, binds with the androgen receptor in the cytoplasm, and when that makes its way to the nucleus, it transcribes certain effects. So for example, if you have DHT, that is going to be in the skeletal muscle converted to a essentially more inactive androgen. DHT does not actually drive muscle protein synthesis, whereas if you have another tissue, take the hair follicle, for example, the scalp, Testosterone is not necessarily as inflammatory or as potent of an inducer of androgenic alopecia as DHT is, whereas we see reliably whenever you inhibit that 5-alpha reductase enzyme, you see improvements in hair growth, reduction in hair fall, things of that nature. So the hormones all have very different effects in different tissues. And you mentioned oxandrolone, which is a DHT derivative. And although it is very similar to DHT, there are some subtle differences in the hormone structure. So whereas if you give somebody loads and loads of DHT, it's not going to have an anabolic effect. Oxandrolone very clearly does have an anabolic effect, and we use this to treat patients in muscle wasting states. Things like major trauma and severe burns come to mind. And it's very clear that that helps them to retain lean body mass and avoid developing muscle wasting in these conditions.
That's very informative. I guess that kind of helps explain some of the phenotypes, like men with 5-alpha reductase deficiency, and also kind of helps explain the difference between just DHT and DHT derivatives. So let's segue into that. Let's talk about the effects other than the androgen binding to the gene. Common question that we get is, what matters more, total testosterone or free testosterone? So there's a lot of effects other than the androgen receptor binding to the androgen. Let's talk about some of those.
Yeah, so you have your total testosterone, and then about 98% of that typically is bound up to sex hormone binding globulin, which is a protein that essentially does just that. It binds up and kind of serves as a delivery regulator and a reservoir for your quote-unquote sex hormones. Mm-hmm. So this preferentially binds to stronger androgens like your DHT, testosterone, and then to a smaller degree things like your estrogens. And things that are highly androgenic tend to decrease the production of this protein in the liver. So like oxandrolone, we've seen a number of case studies, if you will, where patients are using this and you see very low levels of sex hormone binding globulin, and then you see the exact opposite whenever, for example, women on oral contraceptives are using something like ethinyl estradiol. They're going to have very high levels of sex hormone binding globulin because the estrogen induces the production of that from the liver, whereas the testosterone will inhibit that production.
Thank you. Let's talk a bit about what is called heat shock proteins, or HSPs. And also let's touch base on what a molecular chaperone is.
Yeah, so a heat shock protein is just that. It's described as a molecular chaperone. And in response to various stressors, heat stress like sauna or exercise, and interestingly, even cold stress like cryotherapy or ice baths, you will have an upregulation of your heat shock proteins. And what these signal to the body is that you have a stressful environment that's going on, and it helps to stabilize different proteins in the cytoplasm and help them to maintain their function, helps the cell to kind of hunker down, if you will. Mm-hmm. And among those is actually androgen receptors. They are stabilized by a number of different heat shock proteins in the cytoplasm until they're acted on by various androgens.
Mm-hmm. What happens if heat shock proteins are too high or too low?
So in general, an upregulation or overexpression of heat shock proteins is a good thing. It appears they have a number of different protective benefits. When you look at the in vitro data, cells that are overexpressing heat shock proteins like heat shock protein 90 or 70, there's tons and tons of different ones, but they tend to have neuroprotective effects and protect cells from various toxins that they use to induce apoptosis. We see less of that happening. However, whenever you look at some different cancer pathologies, when they're actually examining these things under the microscope, many times those are overexpressing heat shock proteins as well. Mm-hmm. So while it's not clear that the heat shock proteins are causal in the development of cancer, it appears that they are at least involved to some degree, or maybe a response to the cellular stress of that dysfunctional cell. So they have been targets for treating cancer. About a decade they've been developing different heat shock protein inhibitors, but so far they haven't panned out. And it's very difficult because you have some heat shock proteins that actually suppress metastases of cancers. And you have some heat shock proteins, the same ones actually, that will actually promote proliferation, which ideally in treating cancer, you don't want either one of those things. You don't want proliferation of cells or metastases of cells. But one interesting finding that they did have in developing these is that when you treat cells with a certain heat shock protein inhibitor, then you actually have a degradation of the androgen receptor. So it stands to reason that something like sauna use may help to stabilize your androgen receptors and be a natural upregulation of that in terms of the quantity present.
Yeah, it's almost like SHBG, except after it's bound to SHBG. The whole discussion with heat shock proteins in cancer also reminds me of testosterone in cancer. So, that some people think they suppress metastases. They're still trying to establish a causal link with prostate cancer, but they know that it can stimulate growth. Let's talk about some of the metabolic effects of phosphodiesterase E5 inhibitors, for example, tadalafil, which is also known as Cialis.
Yeah, so Cialis, Viagra, the generics now are phosphodiesterase inhibitors, and they have a known effect of improving blood flow specifically to facilitate erectile function. But they also improve blood flow everywhere, essentially, because nitric oxide is a very potent vasodilator, and you're going to end up with more nitric oxide synthesis when somebody is taking something like this. But there are a number of interesting interactions we see whenever you're looking at things besides improving erection quality. So metrics like your insulin sensitivity, metrics like lean body mass improving, body fat mass decreasing, and not to an incredibly high effect. Obviously, something like metformin is going to be a much more potent insulin sensitizer, but it is very interesting. And when you try to dig in and see what is the mechanism, what exactly is causing this, others have speculated that this is due to tadalafil's action on the androgen receptor and potentially its action with the aromatase enzyme. So we do see a improvement, if you will, meaning a higher level of testosterone, lower level of estradiol. And again, it's not a huge jump, but it is something that's there. And we do have in vitro data, meaning in a lab dish, where tadalafil is able to increase the androgen receptor content of cells, skeletal muscle cells.
Mm-hmm. Let's talk about some of the genomic effects of tadalafil or phosphodiesterase E5 inhibitors.
Yeah, so some of the genomic effects would be that upregulation of the androgen receptors, as we just mentioned, and then there's also interesting data with potentially altering expression of genes that could influence Alzheimer's disease. There was a really interesting study published earlier this year where Viagra use was associated with about a 70% reduction in the incidence of Alzheimer's. So we don't know exactly what those interactions are there, or whether there are some confounders, such as people that are using this are healthier at baseline, healthy enough to have sexual activity. But there's certainly some unknown things there, and it's interesting targets for future research.
Some of my favorite studies on PDE5s is the one with Viagra and eyebrow hair growth. And then I also really like the study with tadalafil and arteriosclerosis, not ASCVD, but the actual sclerosis of the artery.
Yeah, the one you're referring to, I believe, looked at the carotids population of older men who were taking alternate day tadalafil, meaning using it every other day, and they saw a regression of the stenosis that was in the carotid arteries. So it would stand to reason that that's leading to better endothelial function, not just specific to the penile artery, but through your carotids. And we do see good correlation between the status of the carotids and the status of the coronaries, both of which are very important for preventing things like heart attacks and strokes.
Definitely. Tell me about the non-genomic effects of androgens.
Yeah, so androgens certainly have genomic effects when they bind in the nucleus and transcribe things. Primarily we think of increasing protein synthesis, but they also have some non-genomic effects that also affect the skeletal muscle, cardiac muscle, One of those factors that is induced by androgens is serum response factor, or SRF. SRF is a protein that, like the heat shock proteins, tend to be downregulated or decrease with age. And this is important for the maintenance of muscle tissue. So when we look at the in vivo data and they induce a downregulation of this specific factor in mice, for the first month they don't see changes, but then after that the muscle loses its ability to repair itself. You see fatty infiltrates and fibrosis and a general deterioration, very similar to what we see over a longer time frame in aging adults.
I think that mostly sums it up. Do you want to comment any on the many varieties of over-the-counter nitric precursors and how they might relate to PDE5s, for example, nitrosogene or arginine or citrulline?
Yeah, so L-Arginine is a very common supplement that people will take to improve their nitric oxide synthesis. But on a milligram per milligram basis, something like L-citrulline is actually much more efficient if that's your end goal. L-Arginine, interestingly, is used to stimulate growth hormone production. So for patients that are undergoing testing for growth hormone deficiency, they may have a arginine growth hormone stimulation test. Some people may be taking arginine before they go into a sauna or before they go to sleep to try and augment those natural pulses of growth hormone that occur. But if you're looking for something over the counter that's going to boost your nitric oxide production because maybe you're fairly young and healthy and you don't need to take something like Viagra or Cialis, then L-citrulline is a good bet to raising your nitric oxide. You do just want to be cautious and watch for a drop in blood pressure, which could precipitate dizziness. So don't take a ton of L-citrulline and then go max out on deadlifts.
Yeah, I kind of see the corollary between metformin and berberine, which is over-the-counter, similar to tadalafil, and over-the-counter nitric precursors like citrulline and arginine. So there's very different mechanisms of action, but people think that they're exactly the same. And the over-the-counter variants are not necessarily perfectly safe for anyone. We see side effects quite commonly. So it's good to chat with your healthcare provider about them.
Yeah, always a good idea to talk about the risks and potential benefits and realize that nothing is risk-free.
Hopefully we've imparted some new information about androgen receptors, about PDE5s, about L-Carnitine, and about androgen receptor sensitivity versus density. Thanks for tuning in.